everybody. Thank you for joining us here today. It's a shame we can't all be together in Manchester like we usually are at this time of the year for MMC, but thank you for coming along to Rob's virtual talk here today. So we'd just like to remind you all that the talk is being recorded. And also there are subtitles or captions available at the bottom of the screen. Do you want to click on the live transcript button? So briefly, I'd just like to introduce Rob before he begins his talk. He's a visual artist and an emeritus professor of arts design and science at Central St. Martin's in London. For the past 20 years, he's worked with botanical scientists and molecular biologists around the world to explore the living world at a macroscopic and a microscopic level. Using a range of complex microscopy processes, he uses a sophisticated coordination of hand, eye, and intuition to create intense, large format photographs that captivate the eye and extend the traditions of botanical art into a contemporary field. His collaborators include the Jordell Laboratory in Kew, the John Innes Centre in Norwich, the MRC in Cambridge, and the Max Planck Institute for Plant Breeding Research in Germany. He works from his studios in both London and Corfu and exhibits and lectures internationally. He's also published extensively, including an award-winning series of books on pollen, seeds, and fruit, in conjunction with Dr. Madeleine Harley and Dr. Wolfgang Stuppi of Kew, and has also exhibitions in North America, Chile, Germany, and Greece, and a feature on the BBC on seeds and climate change. His current research with Dr. Louise Hughes at Oxford Instruments uses energy dispersive spectrometry to record pollutant particles on leaf samples. In 2010, Professor Kesseler was Year of Biodiversity Fellow at the IGC in Portugal, and he is a Fellow of the Linnean Society and an ambassador for the RMS. So it's my great pleasure to hand over to Rob for his presentation, and we look forward to seeing some chat, questions in the chat, and also at the end of the session we will take questions in person too. Okay, thank you, Rob, over to you.
Thanks very much, Kerry, for that kind introduction, and welcome everybody, wherever you are. I'd like to dedicate this talk today to the memory of the late Chris Hawes, whom it was my good fortune to get to know when he was Professor of Plant Cell Biology at Oxford Brookes University. He was a great enthusiast for expanding audiences, and is one of the reasons I'm here today. Over the past decade, collaborations between scientists and artists have become increasingly popular, and sci-art's something of a buzzword. But moving beyond their illustration communication role, there is an appreciation that artists bring a whole new bag of conceptual and technological approaches that open up unexpected and meaningful dialogues, reaching audiences beyond the limitations of scientific journals. And today I want to tell you about the current cross-disciplinary art and science project I'm working on with Louise Hughes at Oxford Instruments that respond to factors impacting on climate change and explores the complementary approaches used by a scientist and an artist in the potential application of colour within scanning electron microscopy and energy dispersive spectroscopy. Before doing that, I thought it's useful to put it into context, and I wanted to show you a few examples of other projects I've worked on over the past 20 years, which hopefully address some of the questions around the benefits of such collaborations. But I think it's fitting to start the talk with two of the greats who exemplify the importance of the image within science. Albrecht Dürer's great piece of turf from 1503 sits right on the border between art and science. It's an acutely observed study of plants, but also a highly selective artistic interpretation of the subject. And of course, Robert Hooke's illustration of the underside of a nettle from his groundbreaking publication, Micrographia. Both of these as topical and relevant today as they were when they were created, really. And still as stunningly kind of beautiful and fascinating. So my work as an artist is driven by the living world and what I see with my eyes and with the help of technology. And I've always been drawn to colour. During lockdown, when I didn't have access to the labs and the equipment I work in, I returned to source, to the plants themselves. And these are from a series of works that explored the staining characteristics of lily pollen, often removed by florists in a form of botanical castration, I think. And the anthers were laid out on wet watercolor paper, and the stain from the oils and the pollen and other anthocyanins in the plant seeped out resulting in a wide spectrum of sumptuous hues. This is a coloured SEM, one of the pollen grains I made almost 20 years ago now for my book on pollen. And under the light microscope, we can see the oily lipids containing the carotenoid responsible for the stain, which is why the florists cut them off. It's a really very, very strong stain. But interestingly, these were first observed under the microscope around 1800 by Franz Bauer, who had a lifetime annuity to document the plants at Kew. And beyond illustration, he was keen to examine their microscopic structures and their functional components using the state-of-the-art microscopes at that time. At the end of the 90s, I had an idea that microscopy offered an unlimited wealth of creative opportunities that no one was exploiting. I approached Kew and was very lucky to be invited in by the palynologist, Dr. Madeline Harley. And she introduced me to her work and the scanning electron microscope. And her images were just, having seen these for the first time, were just sensational. And she not only invited me in, but she trained me on using the microscope. And very soon I was producing my own images of pollen and seeds and plant structures. I think just to observe something so small in such super high definition was truly inspiring. And a privilege in a way. It's not easy for artists to get access to these technologies and to understand how they work. But I immersed myself in it in a way, trying to learn as much as I could and to try and, not to become a scientist, but to try and understand their languages and to try and go as close as I could to their world. And I immersed myself in the complex ornamental coatings of pollen. This is from Amplipracum, a wild leek. Just these fantastic kind of regular, almost hexagonal kind of dimples across the surface. And these are seeds from the same plant. And what's interesting is looking at the surface of the seeds, these interlocking kind of shapes, these jigsaw pattern shapes in a way, which kind of move and flex due to kind of turgor pressure as the cells grow, pressing and giving way against each other. Really kind of fantastic kind of patterns at such a small scale. And of course, architectural structures of seeds. Really, architects and artists have been inspired by plants for centuries, really. And these are almost like 3D architectural models. You can see the layers of cellulose being built up there, almost like a 3D print. And this is a balloon seed, which has it looks like it's a packaging to protect the seed, but actually, when it has a membrane inside that honeycomb, it gives it a bigger wind profile to make it carry further on the wind. Airborne dispersal comes in many guises, and these are pollen grains caught on the fibers of the pollen filter from my old Volvo. These are airborne allergens. The lazy way to collect your pollen samples, I think, drive around a field of dried plants. Around this time in the kind of early 2000s, the use of color within SEM was limited, referred to as false color. And it was really governed by scientific protocols. But I felt that by introducing color, it could be possible to communicate other layers of meaning and reach much wider audiences beyond scientific journals. My color is not false as such. It's based on the original plant. I'll use it to bring out the functional characteristics and structures of the specimen. And importantly, my own response as an artist to the character of the specimen. But they're my colors. And what I mean by that is that I'm not dictated by the program or the protocols within science. After all, no one asked Monet if his kind of water lily paintings were false color. So just as the plant used color to attract an audience of insect collaborators, I use color to attract you. A lot's changed since the early days. as witnessed by a recent advert I saw on the RMS website for a course on enhancing your images to make them even more beautiful and inspiring. I know the art would have some kind of questions about what is meant by beautiful, but I think I know what they mean. But this is in part driven by the advances of technology, which have really advanced so much over the last 20 years. What is possible visually is just sensational. It's also driven by the nature, the competition to get your image on the front of a journal. But perhaps also it unlocks the creative spirit of the scientist. Arising out of this was an award-winning series of books that I did. The first one on pollen with Madeline, and then seeds and fruit with Wolfgang Stuppy. At the time, no one really was interested to publish them. They liked the images, but they couldn't see an audience. I think very little like that existed then. Well, that was almost 20 years ago, and they're all still in print in about 10 languages now, I think. And that gave me an audience across the world that I could never have got so easily through exhibiting work. And Madeline and I, when the first book, felt that, it was really, the images are so startling that it would attract many different kinds of audiences. And so it's proven. Beyond kind of popular science magazines, Guardian, In Focus, and many magazines in languages I can't read, it was also used by some of the kind of scientific communities that I work with. So this was the front cover of the Science Strategy for Q, 2015 to 2020. And I think, to get one's work appreciated and acknowledged across one's discipline, in a discipline one's not kind of necessarily known for, is it gives one a huge kind of, well, it feels great for a start, but also it gives it, as a level of respect which comes back from that. And it also enables me to continue to develop. It gives me a license to continue to develop projects with other institutions, with other scientists, which has been a fantastic privilege. I was fortunate to be awarded a fellowship from Nesta, the National Endowment for Science, Technology and the Arts, which gave me time to explore plant function in greater depth. So these are Medicargo seeds, all from the same family, and with similar characteristics, like the spiral form of the pod, but each with its own individual identity. And his final image of the Medicargo arborea seed won the RMS Scientific Imaging Competition in 2014. To achieve this as an outsider was humbling in a way, and I have Chris Hawes, I think, to thank for some of that. I know he was a great supporter of my work, but he did tell me I was only going to win it once and he wasn't going to let me go in it again. I had too much of an advantage. As an artist, I'm perhaps not tied to the confines of funding bids in the way that scientists sometimes are, or the narrow avenues of research. Narrow, but obviously very specialized. I can tend to meander around a little bit. So these are some ribwort plantain plants, 2 different kinds. And what I was doing on the SM at Kew, I was pushing bigger and bigger specimens into the chamber and photographing them bit by bit across the surface so that I could create a greater sense of physicality and presence within the specimen. And the color is very modulated. As you can see, it's kind of tonally adjusted. I'm not changing the structures. I'm not trying to change it into something that it isn't. But clearly, I am really just trying to light the subject as a painter might across the surface of the floret in this particular case. And it's to achieve this, I take multiple frames at high resolution. So this is an Astrodoll flower, which was quite big, really. It's about 1 centimeter across. And I think maybe took about 80 frames to construct something which I spent a long time piecing it together, working on the kind of the different tonal values so I can really model it in a three-dimensional way to really kind of make this form stand out. And then to introduce color, kind of the final thing. It's quite an interesting challenge actually to do something, a white plant within a kind of digital environment. It was an interesting challenge how to do that, to convey that kind of sense for the flower. And it has a kind of disarming sense of reality. It's real, but it's also slightly alien. It has kind of an interesting edge to it. To really appreciate the results, they need to be seen on a large scale. So sometimes I might project these, but in this case, this is from an exhibition 2 years ago in Greece, and they were printed onto canvas, large format prints hung directly on the wall, so that people could really walk up to them. They were not framed, there was no glass in front, and the black is very intense. It's kind of, which has an interesting spatial quality. So the image appears to float in this dense black hole it's sitting in, floating in. But also what this brought to mind were the 19th century botanical wall charts, which, if you're not familiar with them, are fantastic. There's a recent book published about them. And I think even as a child, I quite like diagrammatic images where they would analyze different parts of a structure. And these kind of stellate hairs are it's very much like an engineering architectural drawing. And it was one from the Hunt Institute of Botanical Documentation in Pittsburgh, who had acquired a whole collection of these, which had really been, I think they've been discarded. They were retrieved from a skip, which says something about the value that is put on things. So maybe they were seen as having little contemporary scientific kind of value, but for me, they have a lot of artistic value. So the museum used it as an opportunity to have an exhibition of these, to clean them up, and to pair up my images alongside of similar subjects, like these stellate hairs. Or in this case, sections through wood. So this is through oak, looking at the very kind of tylose cellular structures in there. And what I like about the kind of wall chart image is that it's, on one hand, it's quite diagrammatic. It looks like an aerial view of something. It could be a beautiful kind of silk print, but it draws you in, and it's fantastically observed. And not just observed and copied, but translated by a draftsman somewhere and an engraver and a colorist. And these are quite big. They're about 80 centimeters to a meter across. So again, you can perceive these from a distance, not just from a screen or from a book. Some of them look like they've come from the kind of notebooks of surrealist artists. So this is kind of some spores there, and my couple of grass pollen grains on the fibers of my pollen filter from the car. Some of them look like very kind of ornate mandalas, something to be kind of entered into, to be mesmerized by, in a sense. And the one on the left is a section through the stem of, it's a garlic, Naples garlic. And this was done on my fellowship with the Gulbenkian Science Institute just outside of Lisbon, 11 years ago now. And the difference there was that instead of working with SEM, I was going to work with light microscopy. And instead of coloring the images post-production, I was staining the samples with histological stains before the kind of specimen went under the microscope. My favorite was the blue you saw there, toluene blue O, which gives a spectacular variety of color responses. So you can see the kind of epidermal cells pink and the chloroplasts floating around inside the cells there in green. And subverting the process again a little bit, I would photograph at the highest mag I could on the light microscope and building up different depths of fields and compressing them till I ended up with a single image. And this image is 4 meters across. Took quite a long time to do, I seem to remember. But it's really, it put an image before the scientists there, something which they were familiar with, but actually had never seen quite before in that way. Also, it was interesting for me the way which different plants, this is a helleborine wild orchid, same stain, but actually different color response through the kind of the chemical constituents, the proteins within the plant section itself. So what I'm doing here, the stain reveals something which was otherwise translucent and very difficult to see. And just being able to have this kind of clarity of the different cell shapes that change across the surface of the section. This one too is about 4 meters across. And they were they had a kind of stained glass-like quality. And we used them to, I wasn't just there to work for myself. I spent a lot of time talking with the scientists there, discussing, looking, how to observe. If you're looking at blood cells every day, how do you identify the one which is the aberrant cell, or how do you catalog? And then how do you present? So it was a fantastic time, and I still keep many friends who are still there. And hopefully I might see this autumn. I've been invited back, so that would be good. Just to show you one of the things that I do with them, this is printed onto silk. It's in the window of a gallery, a small gallery I run in Kennington, just across the river from Tate Britain, and above which I am now speaking from, so where I live. The silk has a fantastic quality, and it lends itself very well to, it's like the window becomes a giant microscope slide. I'm jumping now to the collaboration with Ulla Neumann at the Max Planck Institute for Plant Breeding Research in Cologne. And as well as being editor of the Journal of Microscopy, she's also head of central microscopy at the Max Planck. And Ulla invited me to work with her and Marianne Prischel, PhD student at that time, to develop material for their annual open day, taking the form of microscopy and print workshops for families and a joint lecture. And the popular press often have a habit of demonizing aspects of scientific experimentation. Mutant strains of barley occur naturally and are not just something that's escaped from the lab. So clear communication is important. And the Institute is surrounded by fields of golden barley, a reminder that the landscape has long been established subject for artists, both as a celebration and an immersion in a vital kind of life form central to existence and survival. So this is one which sort of sprang to mind when I was looking at these images of this kind of wheat field by Van Gogh. Barley is the subject of Ulla's research as well, and this is a transmission electron microscope image of a section through one of the anthers of barley. And it had very beautiful kind of soft tones, rather like an etching. And we were talking about how to combine our two works together. So this is, we made a series of works. This is just kind of one of those. So I was trying to really place her images. I was introducing color to her images and trying to place it back within the context from which it came. Images of art and science might be created for different purposes, but have a habit of echoing and anticipating each other. The abstract gestures and chaos and the compression of space within the paintings of Jackson Pollock might be compared with the dried barley stalks in the field. And again, these gestures appear within this TEM image. It's like a kind of a charcoal sketch or a kind of pencil drawing. And I subsequently tried to place that back within its context again, sliding it back into the wheat field. So you're looking at two orders of magnification within the same image. Different orders of magnification are explored further using, this is a stained section of barley anther, and using it in conjunction with cyanotype. Cyanotypes are one of the earliest forms of photography developed by Mary Atkins to record plants in which a solution of potassium ferric cyanide and ammonium citrate are painted onto paper, on top of which a plant sample is placed and exposed to sunlight. The sun fixes the stain, leaving a negative white image when washed out. When the two are overlaid together, we have an image which fuses the two forms of botanical image making within a continuous surface. And apart from the workshops, we extended the reach of our work through poster presentations and through a journal article in focus for the RMS. Reaching audiences is something the BBC are very good at. And this is a project I worked on with Matthew Tucker and Q for an article he wrote about plants suited to climate change. Scientists predict that we're on course for a global surface temperature to exceed an increase of 1.5 degrees centigrade very quickly. And it's important to understand which plants can withstand this. And he understood the value of the high impact images in the service of communicating serious issues can have. So these are wild oats. They have a useful genetic traits, more resilient to drought than commercially grown varieties. It may be inedible for humans, but it is the ancestor of the staple food, the food staple oat, which has been feeding people for thousands of years. The plant species is resistant to a number of diseases that can strike down cultivated crops. So the seeds become very important in understanding what their properties might be. So this is a single seed looked at under the microscope. It's like an intricately crafted piece of jewelry, really, or a magic bullet, perhaps. Like the wild oat, Dorcas carota, wild carrot is a relative of the carrots that we eat and also has these traits which can better withstand drought and warmer temperatures and can be used to develop more resistant varieties. And each seed carries its unique genetic data, which can give us the clues of how we might move forward. And finally, this group, a pair of seeds from Medicare Sativa, which are very kind of ordinary when you look at them in your hand. And I was trying to create something which had, I mean, the Medicare Sativa is particularly resilient to drought, and it's an important as a crop in desert areas in northern Chile, South America. So in producing the image, I was trying to imbue it with the presence of some ancient South American golden treasure. We might consider the seed itself as part of a golden hoard. So coming to Airborne, my current project. And throughout my career, I've been so lucky, really, to work with many different people, experts in their field, who've given their time and their enthusiasm and enabled me to realize the different projects. So this is Louise Hughes from Oxford Instruments, whom I first met when she was running the microscopy lab at Oxford Brookes under Chris Hawes. And we met up again a couple of years ago when she was presenting at the International Congress of Botanical Microscopy. And she introduced me to multicolor electron microscopy and EDS, energy dispersive X-ray spectrometry. And she knew I'd be interested in what it can do. And when she showed me images like this, which is the detail of pollutant particles on the leaf surface, she knew I would be kind of hooked. EDS operates within a transmission and scanning electron microscopes, and it detects X-rays emitted from the sample by measuring the energy of characteristic X-rays. And we can identify which elements are present in the sample. And my knowledge of the technology is limited. And so I'm using this slide of Louise's and her little explanation to explain what's happening. So the secondary electrons that were once atomic electrons that have been excited and have left the atom, they leave a hole in the atom, and the orbiting electrons will release energy in order to jump down the layers of the atom and fill the hole. The energy they release is often in the form of an X-ray. And because the energy layers in the atomic are specific to each element, we can identify the element, and an X-ray has been produced by detecting its energy. Sorry, that's quite a mouthful, even reading it verbatim. I'm not sure if you got that, but it's on the right, you can see this image. So I'm going to scan through different versions of it. And what's fascinating for me is which the individual colors can be assigned to individual elements. So we started with a grayscale SEM. And you can see the color markers at the bottom. So we've got silica, aluminium, magnesium, calcium,
and also using
and the colors can be changed and combined for a final compilation mixed together with the SEM image. So now the scientist has aesthetic choices to make. How do they make a choice of assigning? Partly it must be to do with clarity of what they're trying to show, but there has to be some kind of aesthetic consideration as well. And when looking at these images, it brought to mind the impact that color research and theory played on the development of 19th and 20th century painting. One such theorist, Michel Chavroy, was a chemist, best known for classical researches he carried out into animal fats, which enabled him to really kind of develop the true nature of soap. And he started out as an assistant at the Jardin des Plantes in Paris before taking over the directorship at the Gobelin Tapestry Works, where he worked to improve dye qualities and also develop theories of simultaneous color contrast. So with simultaneous color contrast, Chavroy understood that the perception of color is influenced by its proximity to other colors, distance and volume. And he realized that it was possible to make two colors look like three, which to have both aesthetic and economic effect in manufacturing the design of carpets, mosaics, maps, clothing, stained glass. And what we see here on the left is a pale green background with a darker green square on top. And opposite the left is the pale green again from the background opposite. Well, that's what you think, but actually it's one and the same color. And as we can see here again, the intensity of that color, that red on the left, is quite astonishing. And it harks across to the red on the right. Those two samples are the same. And I think this EDS sample kind of carries that knowledge a little bit further. More significantly, Chavroy's theories were to have a profound influence on the development of painting at the time, and particularly the neo-impressionist painters like the pointillist Georges Seurat, who developed the style of applying color in small dots that from a distance fused to create a more unified surface. And from a distance, the tones became even more unified. And also this unfinished painting by Cézanne reminds us that there are many phases in the development of a painting, phases which are partially hidden from the final result, except perhaps for conservatives who use X-ray photography to peer back through the layers. But in a way, as it is with painting, so it is with microscopy and EDS. Here we're looking at hyphae, bacterial growth on the surface of a leaf. And by selecting the different combinations of assigned colors, and with or without overlaying them on an SEM image, the results are radically different. And nitrogen being particularly strong coming through here into the final image. Really quite a day glow, startling image. So that color does say something, which it's an electric color, which has associations which are almost hazardous. So that's what I mean about having aesthetic choices and the consequences of using particular colors. So from the tapestry weaver's stitch to the mosaic tile to the painter's paintbrush, we now move to the pixel within a digital space, exploring landscape at a different order of magnitude. It's kind of micro landscapes. So this is a backscattered SEM image of an ilex leaf collected in central London, which is heavily coated with a mixture of fungal material and airborne pollutants. Secondary electrons give a more sophisticated sense of lighting to the complex surface. And And just color markers on their own within the histogram adjusted to eliminate noise and a smoothing filter applied. It shows aluminium, magnesium, calcium, silica, iron. And here it's overlaid on the top of an SEM with the addition of carbon in green. Quite a powerful image, quite strong, quite a brutal image. This scan has had the noise reduction turned off and overlaid on top of the EM. The carbon is blue here, so there's a change in character. And with a bit less noise reduction, it comes closer to a pointillist kind of painting by Seurat in a way, which is why it made me think of it in the first place. So here the carbon is green, but now the carbon assigned red, the smoothing filter applied on top of the SEM image. And this is from a series of images Louise produced, and they show something of the color options available in EDS and how they radically change the nature of the image and what is showing and communicating. The high chroma radioactive cocktail of ice cream colors here make it dangerously appealing. And Louise aims to create a palette that is attractive and informative, choosing colors that complement each other while still containing enough contrast for the difference within the layered maps, which can be easily visualized. And the resulting images are not just without interpretation and artistic insight. It's a combination of those. And by adjusting the final composition, she can convey her interpretation of the sample and the data far faster than she can describe in words. I was keen to create a different version that reflected the darker implications of pollution. So I started by using the evidence of each individual element as the basis for hand coloring a grayscale SEM. So it's a grayscale FEM, and then just blending in slowly to create something which was more akin to the disturbing landscapes of Bruegel or the dystopian crystallized terrains portrayed in the novels of J.G. Ballard. So quite different interpretations of the same subject there. It's great to see them side by side. Finally, just to finish, we're doing okay for time, it's good. It's a sequence of images to show how we've been developing the project, using samples collected from sites across Europe. So the specimen we're just looking at was an ilex leaf collected from outside the Garden Museum in Lambeth, on the banks of the Thames, adjacent to a very busy junction. And as you can see, they were quite shiny, but there are also very dirty leaves there. And highly polluted, it revealed spherical forms, possibly aluminium, as well as cuboid blocks caught amongst the hairs of the leaf, possibly a salt crystal. But without EDS, it's not possible for me to know. So this is moving to Padua, the Botanic Garden in Padua. It's the oldest botanic garden in the world, founded in 1545. And beneath this structure is a palm planted in 1585 and still alive, a very special tree. It's doing okay, it's doing okay. And this is the work of another polymath. So this is color theories developed by Wolfgang Goethe. He was both a highly influential color theorist to people like Turner. He also wrote on plant science, and he developed theories of metamorphosis within plant. And he visited there in, when was it? 1786. So I'm not quite sure what he would have made from this sample, which was taken from that tree. It's kind of a cylindrical object from the surface. I'm really just trying to show these connections between artist and science, and artists who are scientists and scientists who are artists. Outside on the ilex, again, you can see highly coated bits of crud. There's a pollen grain there. And a mixture here of oxygen and calcium, silica carbon, It's got magnesium, phosphor. And then my version based on the kind of original data. This is Lake Akrata, high up in the Peloponnese in southern Greece. I was collecting there. I collected ilex really because they don't wither up and dry out. You know, they're fairly solid, and pine needles too. But also for the ideas of filtration and breathing. And here you can see the sample is pretty clean. There are some little bits on the surface, bits of aluminium, silica. And this is the kind of waxy fibrils around the stomatal cell, which are pretty clean, a little bit of crud there. But moving to the the Italian Alps, this is, I think, another pine tree from La Salle. You can see that the needle is dirtier all along the ridges where the stomatal cells were. A lot more built up material there, and the stomatal cells quite clogged. And finally here to Botanic Garden in Geneva, right next to a kind of 4-lane highway in the center of town, completely coated with all manner of pollen grains, fungal spores, and other kind of metals. These are some images of that surface of Louise's, and I just really enjoy the different ways of it is very much a kind of screen print quality, these kind of flat colors. Smoothing gives a different character, and then by changing the kind of assignment of the colors to the elements, it changes again. And then finally to my version based on the data. I started this talk with an image from Robert Hooke's Micrographia, and in 1654 the diarist John Evelyn was writing about plans for a magnetical, mechanical and optics school in Oxford where Hooke was working, and he refers to an elaboratory, a place suitable for scientific experiments. And I think an laboratory or even an e-laboratory would seem like a useful metaphor for the collaboration with Louise, a place to experiment and elaborate on our findings and make a very small contribution to a monumental history. I think I'm going to stop there.
Rob, thank you for such a wonderful talk. That was absolutely beguiling. I'm truly fascinated. Do we have any questions from our audience here today? Would anybody like to ask a question of Rob? If you do, you can switch on your camera and your mic. No? Well, maybe then if I might, Rob, I might just ask a quick one. So you talk about a dystopian color palette. Maybe could you elaborate a little bit more on that? I found that notion really interesting. How would you define that dystopian color palette with regards to, let's say, some of the other color choices that you've made?
I think it's something to do with the electric nature of the colors that Louise was using. Dystopian more, it's the way in which languages build up around how landscape and our environment is described, and the way we're either alienated from it or drawn into it. And it's, I was thinking in particular, I think I can't remember the name of his book now, one of it's kind of Crystal Worlds. He conjures up this really fascinating kind of landscapes where it's changing. And the Bruegel's kind of landscapes as well. There are sort of these landscapes in turmoil. Many of his have people in them as well. So it's like a small excerpt from something that was happening within that kind of landscape.
So we're all clearly living in a dystopian world at the moment. I don't think any of us thought 18 months ago, you know, that we would really be here still, but or here at all, really, for that matter. But I think when you see this data presented in this way, it's just really, really wonderful. So somebody has just posted a question. It was wonderful to hear about the sharing of knowledge in the labs. I realize I'm not sure if you are hand coloring the images that you transfer to Silk, or is it all done on computer? So that's from Nikki O'Donnell.
Okay, Nikki. Hello, Nikki. It was all done, when I say hand coloring, I use that term very deliberately, and it was deliberately to distance itself from false color. It's all done digitally within a digital environment using a graphic pen on a tablet. But when I say it's hand colored, I build up multiple layers of different colors and erase back through that. And the process is no different to when I used to do large format pastel drawings or watercolors. The sensitivity and the sensibility is really the same. Into the silk, that's digital print onto Japanese silk, particular kind of Japanese silk, which the dye goes all the way through. So it's really impregnated into the surface. Yeah, maybe I could try hand painting silk. It's another. I think I need about 10 lives to kind of get through all the kind of possibilities that come up in one's lifetime, really.
Yeah, no, I could imagine that.
You just muted yourself, Nikki.
Muted. Oh, I'm muted. Sorry, it keeps flicking one way or the other. You know, I realized I hadn't questioned, you know, the different formats, the silk or the photographic or the digital, but yeah, so it's all digital, but the way you apply it obviously depends on the material.
Yeah. I mean, I haven't shown any today, but I I have done a series of drawings using Indian ink mixed with these dyes. That's how I started doing it, and then mixing with aniline dyes and staining the paper. So there is an overlap, and that's why I started doing those pollen prints in the winter. And I know that Louise has got some ideas in the pipeline for how we might develop that, really.
So that's actually leading, Rob, into Louise's just asked, where would you like to go next with the project?
Okay. Well, I actually wrote something. So I said, I suppose the question is where we go next. So, I mean, thinking back to the origins of Chavroy's work in tapestry, you know, we both felt that these images are really tactile. I mean, they could be like fluff deposits on the inside of a Hoover bag, or kind of very rich tapestries. So I think tapestry is one way we want to look at, and whether it's digital or whether it's kind of manual, both are quite complex and probably expensive and highly time consuming, but we'll explore that. And I think the other was what I was saying about looking at plant color, and what does that, you know, I'm quite curious to look like. Actually, Louise, I think I sent one up to the lab, one of the kind of watercolor printouts, so we can look at that in EDS and see how those colors match up to the colors that the plant itself has.
Yeah, I think that's so interesting. So Johanna here has asked as well, from your work, do you have any advice for scientists on how to treat color when they are selecting colors for overlays in fluorescence or in EDX?
Fluorescence. I mean, okay. When I went to the AGC, I was hoping to work within confocal microscopy and fluorescence, and actually, it was just so competitive to get onto the kind of confocal. I never had enough time to. I learned how to use it basically, but I needed kind of a couple of months to just manipulate it my way. But since then, I mean, the stuff I see coming through online is just sensational, really. But it is quite hard to personalize it, I think. I mean, I think the technology is doing so much for you. I think you have to stand back a bit and you have to think, what am I trying to communicate here? What is happening in that subject? And also to try some different variations, you know, and test them out on people, to try different, similar and radically different combinations to see what that does to the form. I know you can, yeah.
There's a lot of talk lately about the representation of colour for colourblind people. So, you know, combinations of reds and greens we traditionally see in fluorescent images, and trying to kind of move away from them to a more, you know, accepting palette, really.
Yeah,
so
that's interesting.
yeah, so somebody, Aideen from NUI Galway, has asked, absolutely fascinating and beautiful. I'll never look at SEM and EDX images in the same way.
Good.
So,
that's the idea, really. I mean, I think one of the things that came out of doing the books that, you know, we felt that, you know, when the publisher said no one, oh wow, you know, it's amazing, but who's going to buy these books, you know? We felt, well, actually loads of people, you know, and there's 20 times more work than I can ever do in here. So it's also a gift and a challenge to other people to take on and do something with this. It's a way of sharing, a way of kind of giving back, and a way of seeing what other people do with it.
Yeah, it's absolutely wonderful. Is there any more questions from our audience? Anyone have anything else they'd like to add? There's really wonderful comments coming in here, Rob. You'll have to have a look through them at the end. Really, really positive. Everyone has really enjoyed your talk, and really, really inspiring. I think definitely for all of us here who have an interest in the art science interface, I think it's really, truly inspirational.
I think
a pleasure to be able to host you here today, and to be able
you.
to have this lovely chat, and to see the newest work that you're doing with Louise. It really is fantastic, and I look forward to seeing more, hopefully, in the future.
Yeah. Well, I think we're meant to be having a journal article come out fairly soon about the work, so that's good. But yeah, you know, it was a bit frustrating. We just got going, and then, wow, 12 months ago, everything got kind of closed down. So hopefully we can sort of start kind of gearing up again.
Excellent. I look forward to it. And with that, we might bring the session to a close, and we thank everyone again for coming along and listening and participating in the talk today. And once again, Rob, thank you very much for a really, really wonderful, inspiring talk.